Chapter 22, Part 2, Screencast 2. Now, this particular screencast will be focusing primarily on T lymphocytes. And remember, they actually cannot directly bind to the antigen, so they are going to require antigen-presenting cells. Antigen presentation, these are cells that specifically display the antigen on a plasma membrane, so T cells are capable of recognizing it. There are two categories of cells that are able to present antigens to T cells. All nucleated cells of the body. So this leaves out red blood cells because remember, they do not have a nucleus. And we also have another class that are specific antigen presenting cells. We'll be calling them APCs moving forward. And these are actual immune cells that present to both helper T cells and cytotoxic T cells. These are going to include dendritic cells, macrophages, and B cells. Now, major histocompatibility complex molecules, or MHCs. Now, this process of presenting an antigen is going to require the attachment of the antigen to a major histocompatibility complex, or an MHC. Now, these are transmembrane proteins, and they actually serve as a docking site for specific components of antigens that are then displayed to T cells. MHC1 is found on all nucleated cells, so remember, not on the surface of red blood cells, and MHC2 is found on APCs, in addition to MHC1. So remember, APCs will express both MHC1 and MHC2. Cytotoxic T cells, they will interact with MHC1, and this is going to be called an endogenous pathway or endogenous antigens. Helper T cells will interact with MHC2 or exogenous antigens, always foreign. Now, how I like to keep in mind how this works, I look at C becoming before H within the alphabet, 1 before 2. So this helps me to remember the cytotoxic T cells interact with MHC1, whereas the helper T cells will interact with MHC2. Now, MHC class 1 molecules, these are glycoproteins, and they are genetically determined structures that are unique to each individual. They are continuously synthesized and modified by rough endoplasmic reticulum, then inserted into the cell membrane. They will display fragments of proteins that were bound in the rough endoplasmic reticulum. If fragments are from endogenous proteins, immune system recognizes them as self and ignores them. If fragments are from an infectious agent, immune system considers the antigen non-self. This is communicated to cytotoxic T cells that they should destroy that cell. Now, the next 3 slides are some really important images that are going to walk us through the process of the formation and docking of MHC. Now, we're first going to look at MHC class 1 molecules, and this is going to be in a healthy cell. Now, what we're starting off with here is going to be these peptide fragments of self-antigens. Now, notice they're going to be entered into a vesicle, and they're going to work their way into the rough endoplasmic reticulum. Now, in the rough endoplasmic reticulum, MHC class 1 molecules are continuously being synthesized. During production, peptide fragments of the cell bind with the MHC class 1 molecules. So what we can see here is that the MHC1 complex and the self-antigen will actually combine and perform or leave as a transport vesicle. This transport vesicle will actually make its way to the Golgi apparatus. It's going to be reprocessed to a further degree. And then, once again, we're going to see the MHC1 with the self-antigen, and it's now in a secretory vesicle. Now, these are vesicles that are capable of inserting the MHC class 1 molecule into the cell membrane. And notice, we now see the self-antigen expressed on the outside of the cell. Now, we're looking at the docking of MHC class 1 molecules in an unhealthy cell. So notice we're starting off with viral particles that are inside the cell. They will interact with a proteasome, and this is actually going to digest the protein of viral particles. Now, these peptide fragments of the viral particles will actually form a vesicle, and within the rough endoplasmic reticulum, they will attach themselves to MHC class I molecules. Remember, MHC class I molecules are synthesized within the rough endoplasmic reticulum. As we saw previously, a transport vesicle is going to be formed, and that contains both the MHC1 class I molecule and the viral peptide fragments. They are going to be shipped through the Golgi apparatus and eventually to the plasma membrane. Once leaving the Golgi apparatus, we see that they are in a secretory vesicle, and once again, that is capable of inserting the MHC class I molecule along with the foreign antigen into the cell membrane. So now, once again, the foreign antigen is on the outside of the cell and can actually be presented to a T cell. Now we are looking at a MHC class II cell. Now, MHC class II molecules are synthesized by the rough endoplasmic reticulum of the antigen-presenting cell. Now, the MHC2 class molecules are shipped by the endomembrane system through the Golgi apparatus and form transport vesicles. During the process of phagocytosis and destruction of an exogenous antigen, vesicles that are containing digested peptide fragments called phagosomes actually combine with a lysosome. This combined structure is a phagolysosome. Vesicles containing MHC class II molecules merge with the phagolysosome containing the digested foreign antigen. The combined structure is now a secretory vesicle, and within this structure, antigen MHC class II molecules bind with the foreign antigen. Eventually, they are going to be inserted into the plasma membrane, and once again, the foreign antigen is expressed on the outside of the cell membrane and can be presented to a T cell. Now here we're looking at the interaction of receptors of T lymphocytes with MHC molecules of other cells. So note what we have here. So here's the antigen-presenting cell. And remember, as an antigen-presenting cell, it has both class I and class II MHCs. Now, if we look at the T lymphocyte, okay, and we're basically looking at the ability as far as what MHC class I will actually bind to, specific to these proteins. Now, CD8, these will be the cytotoxic T cells, and remember, they are going to bind with MHC class I. And then if we look at the CD4, or the helper T cells, they actually are binding with the MHC class II antigen on the specific lymphocyte. Now, a topic that is related to this is going to be organ transplants. And when someone is receiving an organ, it is important that when we look at the donor, they're not just matching blood type. It goes far beyond that. So it is part of it, but what is really necessary prior to donation is a good match with MHC antigens. No two individuals have exactly the same MHC molecules, so generally the best chance for a transplant is a first-class relative. They probably will have closer MHC complexes. Now, remember, these are components of the innate and adaptive immune system, and the issue here is the attempt to destroy transplanted tissue. And the problem here now, anyone who is getting a organ transplanted, they actually will be taking immunosuppressant drugs the rest of their lives while they have that transplanted organ to prevent organ rejection.